It's great to be here, and I'm happy to announce that this is like my third birthday in a row without an AlphaFold release. So I'm very happy that today I don't have to worry about structures. So I wanted to start by not using the usual plot showing you the sequence structure gap. So I decided to come up with something different, show you the protein universe. So think about that little dot right there as protein structure and what we know about protein structure compared to a sea of black that is protein sequences. Over time, that little dot grew in size. Thank you to the protein database. And using information encoding in the protein data bank, we've been able to annotate a lot of different sequences that are available in other resources. But just to give you an idea how many sequences we have, I decided to make this visualization. There's lots of other ways to annotate structure, but this one I think is good as a visual way to do it because the radii of all the circles you're going to see, they're all proportional to the size of the data sets we're analyzing. So PDB, as of literally this morning, has 239,000 structures. And that is just a small subset of all experimentally characterized sequences available that are marked as experimentally characterized in UniProt. And this is 2.5 million sequences. But once you start looking at all the sequences that we're not able to characterize experimentally or they haven't been done yet, when we move to computationally annotated sequences, this is just a small portion of all the sequences annotated in UniProt, just 250 million. And once you start taking into account sequences now that are coming in from metagenomic resources such as Magnify, it's just a little speck, even in this case. So if we have an overview here, very similarly to the actual universe, we do have a different pace of expansion. We are accumulating sequences at a pace that we're not able to characterize structurally. So luckily, this changed a couple of years ago now when AlphaFold was introduced, and thanks to an embedding-based predictor based on made by Meta called ESMFold, we were able also to obtain structures for the whole of ESM in a resource called ESM Atlas. So in collaboration with the group of David Jones and Martin Steineger, the University of Seoul, we wanted to look at what part of the structure space is unique to ESM Atlas. So we created this data set called AF-ESM, where we put together all 606 million protein models obtained from our ESMFold in our ESM Atlas. We put them together with AFDB, the whole of FDB, and we created AF-ESM, which is near 1 billion structures, basically. So the way we wanted to tackle this project is in 4 parts. So the first one, we were really interested in looking at, do we have certain specific structures that are unique to certain biomes that are available in ESM? We wanted to annotate individual structural proteomes. And then we wanted to see, do we have novel domains, novel folds that they haven't been seen before in AFDB? And do we have some novel domain compositions inside each individual chains? So all of these 4 tasks basically have this task in common, which is we started by concatenating and clustering the whole data set. So we started with this 821 million structures. We clustered them by sequence using MM6. We obtained 187 million sequence clusters, and then we retrieved what we thought was the best representative for it, using a minimum coverage of 0.7 across the cluster and a minimum PLDDT over 60. And this brought us down to 102 million. Then once we obtained those structures, we clustered them using fold-seq cluster, using an e-value of 0.1, 0.01, and a 90% coverage bidirectional. And we ultimately ended up with AF-ESM, which basically consists of 72 million structures that we carried for our analysis. So one of the things we were interested in, as I mentioned, is since we have organisms that they adapt to extreme environments, do we have any extreme environment-specific proteins? Luckily, data coming from ESM Atlas comes originally from Magnify, and we have biome annotations for most of them. And what we did, we decided to calculate what is the lowest common ancestor for a biome. So for all the 72 million, we looked at clusters that had at least 10 biome annotations for each proteins within them. And for example, here you can see you have root, environmental, and then it differs at the leaf. So in that case, the lowest common ancestor is environmental. So one thing we noticed is that out of those clusters, we do have 1.5 million proteins that are universal. We can find them across all biomes. And then we have unique clusters that are spread around. We have certain O-specific ones. We have 15,000 that are associated to human. And then we can whittle down all the way down to the ones we were interested in the extreme environments. One thing we noticed, though, is that these annotations, they tend to be really sparse when we looked at taxonomy. So we decided to do the same thing at the taxonomic level. We applied the same concept by taking all of our 600 million sequences. We scanned them against the genome taxonomy database, combined with UNIREF90, and we were able to annotate 423 million sequences in ESM Atlas. So you can see that we have this information for 222 million species that before were unannotated. And we can give an LCA of for 2.3 million sequences for clusters at the super kingdom level. And by combining this information at the biome and at the taxonomy levels, we're able to find some protein structures that are unique to certain specific environments. For example, we found some proteins that are uniquely associated to hyperthermal environments, some certain proteins belonging to thermoproteales. We have DNA primases, thermopsins. Then we started looking as well as proteins that live from organisms from hypersaline environments. So we would expect a bit more kind of flexibility in order to adapt to these saline conditions. And as well, we looked at proteins that are unique to certain glacier conditions, so really low temperatures. And we found some proteins associated mostly to cyanobacteria, caspase domains, or nucleases that are specific to these environments. But this was done at the chain level. And one of the things we were interested in, as you can see, these are chains that are multi-domains. So we wanted to replicate some of the analysis we did in TED, which was our approach to classify every single domain in AlphaFold database. So we applied the same approach we used for that project by doing a domain analysis of the ESM unique part of AF-ESM. So for those of you that are not familiar, we did this project last year. We took the whole of AlphaFold database, 240 million chains. We chopped them up into constituent domains using a consensus approach. Then we clustered both at the sequence and structure level. Most of them mapped to CATH and PDB-associated structures. And then for those things that we were not able to map, then we were able to put them into a series, several steps of clustering and searches against existing databases. And we identified 7,000 novel folds that are single domains and 6,400 folds that we don't know if they're still novel, but they're highly symmetric. They're really beautiful on T-shirts and posters and mugs. So we decided to apply exactly the same protocol to this data set from AF-ESM, hoping to find novel folds that are unique to bacteria. So what we did is we took the 3.5 million ESM-only proteins. We applied our consensus chain segmentations on 3.2 domain-containing chains. We obtained 5 million domains in this case. And then we put them through several steps of homology search and quality assessment. We noticed that 50% of them already mapped to a domain already classified in CATH. But then something strange happened compared to AlphaFold, domains that we analyzed in AlphaFold. We lost a really big amount to quality during the quality assessment stage. So we only end up with 183,000 domains at that step. The next step We went by doing iterative clustering, and we searched them against existing databases, the latest release of PDB, just in case somebody crystallized them or characterized them in the meantime. And these numbers, they slowly start whittling down as we go. As the next step, then we looked at if we had actually domains with internal symmetry. So we ran them using ISMB, and we clustered them. We ran them also with using a deep learning-based predictor for quality called fold class DOM-QUAL. And we ended up with 7,000 cluster representatives. And as a final step, we clustered them with the whole of Cath, ECOD, SCOP, and SCOP-E, as well as PDB, and we stripped out any domain that was already classified in these resources. So at the end of this process, how many novel folds do you think we have? 0. We were very disappointed, but also excited because, I mean, it's a result in itself because, I mean, it's a massive data set, 3 billion structures, and we have nothing novel. Why? I think the main culprit is this, where we discarded 92% of domains, which indicates that probably a lot of ESM fold predictions are not really well made, and potentially something that we cannot model. Maybe they are something really, something we can remodel potentially. So should we go ahead and remodel 2.3 million domains with AlphaFold? Why not? We did that. Still no new novel folds. So we then in the end, okay, why? We started looking at the composition of these errors, and there's a lot of fragments, a lot of bad gene calls, but we started from 3 million chains that are unique. So why? Something novel must be there because they're not in AlphaFold database. So I think what we are looking at is that we do have, for the things that we're able to characterize, we do have the same domain toolbox, but in different arrangements. So we went back to the chains and we started looking at which one of them are multi-domain chains. We have 400,000 of them. We extracted individual and unique domain combinations, so A, B, C, or B, C, A in the chain, for example. And we found there are 4,000 novel combinations that are not classified in AlphaFold database. So that actually covers only 3% of the data, though. So there's a lot of data that is still uncharacterized. One thing we realized is that a lot of this novel multi-domain characteristics, multi-domain combinations, they are associated with receptors, particularly. For example, these TOMB dependent receptor plugs, you have these AST likes, and they all are always in different combinations with other domains. For example, these are all novel combinations that you cannot find in AlphaFold database. So the issue is we have a lot of structural novelties that are really elusive to current AI-based methods. So I think this is an initial look at the data. Doesn't mean that there's no structural novelty in metagenomes, but this is what we found in ESM Atlas. So this is one way to put it. So all this data that we analyzed is available on afsm.foldseek.com. Here you can put your UniProt ID, your Magnify. You can see specific cases that we isolated that we thought were interesting. And you can explore each individual cluster. You can look at representative structures, all the taxonomy annotations that we made. And if you want to actually download, if you want to read more about it, you can read our preprint. At the moment is under review. And all of our data and code is available on Zenodo and dedicated websites and on GitHub. And with this, I would like to thank everybody that was involved, particularly the team of David Jones, Andy Lau, Sean Gambethil, and everybody in the team of Martin, particularly Jingi and Yuan, that did most of the work on the multi-domain analysis. And with this, well, thank you also for your attention.
Thanks, Nico. So, any questions?
Can this lack of diversity or novelty be resulted from just how ESM fold, RunFold are predicting structure? Like maybe there are actually no structures, but then just because of how these models are trained, they only predict structures composed of fragments of known structures.
Exactly. I think one of the intrinsic limitations of all the predictors that we have at the moment is that they perform really well because they have been trained on PDB structures. But the more you move further away from things that are not in PDB, the worse they get. So it's kind of a dog chasing its tails in the end. So it's hard to predict something that is not in PDB if we keep training on things that are in PDB. Yeah.
Happy birthday.
Thank you.
Okay, you found 0 faults, but you included millions of new sequences. I wonder, have you calculated the delta entropy per fold in the sequence space? Do you understand what I mean?
I do, but we didn't.
Okay, that would be really cool.
Yeah, I mean, I think also a lot of interesting stuff is in between. You know, we have things that we map already to things that are in PDB, and then the novel faults that we couldn't find. So I think there is a lot of gray area, that these are remote homologs of things that are already classified in homologous superfamilies in Cath, SCOP, PCOD, and potentially new functions in there as well that they evolved over time. But that's not something that we looked at, and it's something that we're going to work on.
We have an online question. Ravi Avrol says, great presentation. Why did you use ESM structures when I think it is known that many of the structures are not high quality?
Well, the main reason is because there's no alternative. So there's no, I mean, ideally in a fantastic world where we can model everything with AlphaFold free, we would just like model all 600 million chains or sequences available in ESM Atlas with something that is a bit more accurate. The big advantage of ESM Fold is that not that it's accurate, but it's extremely fast. It's like takes 1.5 seconds to predict the chain, the structure for a chain. One thing also, there's a big caveat here. We're also looking at clusters, and there's a lot of variability inside these clusters. Because even when we looked at TED structures, where across AlphaFold database, we can find structures for things that are 100% sequence identical, and we do have dramatic changes in random SDs. So it's in the end, I think we just looked at a snippet of all this data that is available right there. In an ideal world, we would have treated every single chain as the same. We would chop everything into domains, making probably 3 billion domains, but that's not what was feasible at the time.
Thank you very much over here for a really great talk and puzzling results. I fully agree. Could you comment a bit more on the quality? You said already that that was probably the biggest thing, that you had to remove all of these structures from your analysis, and you said because they were poor quality.
Yes.
Could you elaborate a little bit what you meant? Was that just like average confidence or something?
It's a combination of a lot of these things. So mostly the PLDDT is lower in general across every structure predicted by ESM Fold. One thing we also noticed is that it tends to, as soon as you move to remote homologs, it tends to perform worse than AlphaFold 2. Its confidence drops off a cliff. So in order to test this, we took all the 7,000 folds that we found in TED that they were modeled with AlphaFold database, and those ones with AlphaFold 2, and those ones, when we selected them, they had a PLDDT over 90. We looked at them physically; they were really well modeled. We remodeled those sequences using ESM Fold, and the quality is dropped to 40, 50 PLDDT. So absolutely rubbish. So that was one thing. Then we also have a lot of fragments in there, things that are potentially not proteins as well. So it's kind of a mixed bag of things that are happening here. We do have a supplementary figure in the preprint where we actually have a breakdown of things that for each category, classified in Cath, not classified in Cath, we discarded it because of disorder, because of this and this and that. So if you're interested in that, it's all tucked away in supplementary.
Thank you.
Okay, thanks.
Next one, we'll have the next person.